Welcome to this lecture on allergic reaction mechanisms and the penicillin-cephalosporin cross-reactivity. At the end of this lecture, you'll be able to identify the features of the immune system related to drug allergies. You'll be able to discuss the major histocompatibility complex molecules and their interactions with T-cell receptors. You'll be able to discuss the issues related to the formation of antibodies against small molecules or drugs, and discuss the factors associated with penicillin and other drug allergies. This is the last slide of the presentation. When we get to it, you'll know the lecture's at its end. And by the time we get to this, the reason we're going to cover everything between this slide and actually seeing it again is I want you to be able to answer questions as to what is a drug hapten? How does it get there? Is this something that's likely to how easy or hard is this? What's the role of the T-cell receptor and the major histocompatibility complex? And what is this antigenic peptide? So, to begin our story, remember that there are two parts to the immune system, the innate immune system and the adaptive immune system. The innate immune system has all sorts of elements in it, including natural killer cells, mast cells, and a variety of cells. And these are non-specific defenses that the body uses. So it will respond to any kind of bacteria attack, say, using this method. Then there's the adaptive or acquired immune system, and it has components that include T cells, B cells, and antibodies. And we see that the adaptive system actually can end up using elements of the innate immune system. This lecture will proceed in three parts. First, we're going to do a review of the immune system such that later on, if you have any gaps in your knowledge or any misunderstandings, you don't have to go looking elsewhere. You can just refer back into the lecture notes and you should find the answers to your questions. So I'll kind of glide through the area on the immune system. Then we're going to talk about the MHC and TCR receptors and how normally antibodies are formed. And then we'll consider then how do drugs fit into this immune system and how do we end up with allergic reactions. So, in understanding the innate and the acquired immune systems, it's useful to look at where do the cells in the immune system come from. This is one of the challenges of immunology, is understanding the different factors, including the different cells that are involved in it. So we see that actually, generally in the bone, there's actually a multipotential stem cell, and it leads to two classes of cells being formed, those which are myeloid and those which we call lymphoid. And you think of the lymphatic system when you think of lymphoid. The myeloid, we see this cell can give rise to red blood cells, which have hemoglobin and can carry oxygen. We have the formation of thrombocytes, which are the blood platelets, which are involved in blood clotting. Then we have mast cells. Notice that they're actually a class of their own. Then we have the myoblasts, which give rise to four types of cells: basophils, neutrophils, eosinophils, and monocytes. And the monocytes, which we'll see are actually the Phagosomes, end up being able to eat other things. They include the macrophages and the dendritic cells. Then over on the lymphoids, we have the natural killer cells, which is part of that innate immune system. And then we have these lymphocytes, which are the T cells and the B cells. And from the B cells, we end up making plasma cells, which make antibodies. So those derived from the myoblasts and the lymphoid progenitor, these are all considered white blood cells. The mast cells actually we'll see are found in tissue, and they're not, and their progenitors are floating in the blood, but they actually form tissue, so they're not normally considered white blood cells. So looking at what are considered white blood cells, we see there are 5 types. We have the lymphocytes included in here, and that's going to be the T cells and the B cells. If we see that, especially involved in allergy reactions, there's going to be the neutrophils and the eosinophils and the basophils are going to be most involved in the responses. We see that neutrophils take up from 55 to 70% of all the white blood cells. And we see these basophils are actually less than 1% of the white blood cells. The basophils can release 2 chemicals, histamine and heparin, which are very indicative of the symptoms that we observe in immune responses. But notice they're a very small percentage of the white blood cells. What we'll see is that mast cells and basophils, these 2, both release histamine and heparin. The others don't. That's not what they're known for. So that's the white blood cells. And so looking at the macrophages, which are derived from the monocytes, these we see have appendages that can go and do things like collect bacteria or other substances, and they can actually engulf them and then digest them. The dendritic cells, which are also from these monocytes, also can, in response to cytokines, they mature, and so then they can go and also engulf and then present on their surface to activate other elements of the immune system. So again, macrophages, dendritic cells, these are, in this class, known as antigen-presenting cells. They can take up things that are foreign in the body, and then try and activate the immune system so that it can resist it. You already start seeing here elements such as, here's a CD4 on a T cell, and these are different receptors, and this makes the different lymphocytes have different selectivities, and you'll be seeing the importance of that. So these mast cells, which are not white blood cells, but like the basophils, release histamine and heparin. Also, what makes them distinctive is that they can get covered. First of all, they circulate in the body as in the blood as precursors, and then they get into different, either into connective tissue or into the mucosa, like the lining of your mouth and lungs. And under the influence of interleukin-3, a chemical, they mature. And once they mature, they're actually a type of antibody called EGE, which actually ends up binding all over the surface. There's a word for that, opsonization, is when a chemical binds all over the surface of something. And then these antibodies then can recognize antigens, and then these mast cells will then undergo a process called degranulation, where they release histamine and heparin into the environment. Looking more closely, these mast cells, which again are a class of their own of cells, have many different types of receptors, and they again can be activated and changed by cytokines. Then there's chemokines, which are that's again part of the innate immune system, where basically the cells can sense chemicals and actually move towards those chemicals. Then you have these antibodies from the IgE, which can recognize allergen and antigens. They have other ways to interact with other cells. So they're quite a complex cell, but the main thing is they release these mediators. And they're involved in many different types of disease states or things that are observed. One of the consequences of mast cell activation can be something known as an anaphylactic reaction. This is a worst case scenario as far as disease states go. So basically you're going to have antigens, okay, that, and we'll talk more about these in a moment, that eventually get recognized by that through the process, which we'll discuss, will result in B cells making antibodies, which then coat these mast cells. And then when the mast cells see these antigens again, then they're going to be releasing the histamine. And when this reaction becomes not extreme, but when it becomes very strong, the heparin, first of all, causes the blood not to clot. The histamine causes the vasodilation and can lead to swelling of tissue. And if that's the airways, you can end up then having trouble breathing. So, and that's why sometimes people carry the EpiPens. It's in case this reaction gets triggered. So again, one can imagine, thinking ahead in this lecture, the antigens are something that's going to cause an allergic reaction. And then if you have too much degranulation, too many mast cells release the histamine, you could have this anaphylaxis, and it could be life-threatening. Other elements of the immune system, particularly the innate immune system, is something called the complement system. There are actually 3 different pathways through which they get activated. So these are not controlled by antibodies. They contain some proteins with rather interesting functions. So there's something called convertase, and they have several in here, convertases in this pathway. But C3 is the most important one. And it's sort of like in the future, or if you were to study viruses, they make long polyprotein precursors, which then are able to cleave themselves up into smaller proteins. And the convertase is like that. It has the ability to self-cleave itself. It has its own protease, which can actually cleave peptide bonds, and it can cut itself up into 2 main proteins. C3a, which increases the permeability of blood vessels, and C3b, which actually, if it associates then with, can actually react with bacterial membranes and tag them for attack by phagocytes, those cells derived from the monocytes, such as the dendrites and the macrophages. So if C3b is attached to a bacteria, it means that it's going to start attracting these macrophages, and they'll go eat them. And then this C3 can also cleave and start processing another protein called C5, and that helps mediate inflammation. There's CC5A rather, and that's attracted very much with anaphylaxis because it causes inflammation. Again, inflammation of the airways, difficulty breathing. There's another component derived from C5. Again, C3 helps C5 get cleaved into C5A and C5B. C5B interacts with some other proteins in this complement cascade, and they together can form pores, which then lead to disruption of bacterial cell membranes. This is kind of a foreshadowing of what we try and do with antibacterial antibiotics, which weaken the cell membrane. Basically, the complement system has a mechanism to do that using these particular drugs. So I've mentioned IgE. Just to refresh, there are a number of types of antibodies. There's IgA, which gets secreted into the gut and into our mouth and our saliva, even in our tears. There's IgD, which is very much associated with this other antibody called IgM. IgM is the first antibody that we see involved in immune responses. It's not connected with making memory cells. Then we'll see IgE, which is this antibody that binds to these mast cells and the basophils. So notice, normally when you're talking about mast cells, which again is not a regular white blood cell because it's localized in tissue, not in the blood, and then basophils, which is located in the blood, but which is the lowest white blood, the least often occurring white blood cell. So you often hear mast cells and basophils talked about together, again, because they both release histamine and both release heparin, which is an anticoagulant. And then there's IgG. This is the main antibody found in the blood. This is the one against which creates memory cells, which then gives you long-term protection. So just looking at the structures of antibodies again, you're going to have basically, they always depict them as a Y shape. We'll see they're actually not quite that way. But you have some antigen binding arms. In other words, these arms, the variable light and the variable heavy chains, will be able to adapt and recognize certain molecular structures. Then these chains are normally bound together by disulfide bonds. And then you're going to have a region called the FC region. And this is the region which actually, it basically is the adapter between a cell recognizing antigen, and then actually the FC region determines what cells or what types of response the immune system will elicit when it recognizes this. So in this case, it's going to be the FC region which binds to those mast cells. And that's why the IgE coats those mast cells, because the FC region is especially adapted to recognize and bind into the surface of the mast cells. So just another recognition. We see here actually, we normally are drawing these chains as being linear, but actually they end up joining each other in IgD and IgE. They're not quite linear. This is how they actually look a little bit. And actually the same is true for the IgM. So normally you have 5 antibodies which are on the IgMs, and they come pre-packaged with the ability to recognize a lot of different molecules. And then, so they first bind to the antigen, and then subsequently there'll be an evolution, and the IgMs will give way and IgG will form. Notice the IgA, it actually acts as a dimer, and over it, it's actually associated with a protein that allows it to be secreted. So both the IgM and the IgA have something in common, and that more than one chain associates with itself. And the IgAs are very important in the gut. So when you have, at a future time when we study things like polio, the virus actually exists in the gut. So if you get a shot, which is since 2000 that's been the case, you make IgG antibodies against the polio virus, and that protects you against polio. But you're able, because the virus actually reproduces in the gut, you're actually able to carry it around. In the early days, back in the 1950s, they first came out with the shot, which allowed the production of IgG. Then within 2 years later, they came out with a vaccine that you then drank. And that, when you ingest the antigens, then you make IgA antibody, and then the people who drank that vaccine against the polio virus actually produced IgA antibodies, and they actually eliminated the virus from their body. So they were not only protected themselves, but they weren't carriers. Around 2000, because the type of vaccine, 1 in a million times you could actually end up getting polio from it, they stopped giving it to people because the risk of catching polio at that time was less. But anyway, the type of antibody, where it is, what is secreted, this is all important. And again, IgE, which is the least common antibody, but it's incredibly important because it binds to mast cells. So we don't need a lot of IgG around, but it is the most common antibody we'll see that's associated with allergic reactions. Just to point out, B cells themselves end up having expressing a number of antibodies on their surface that they get activated through the mediation of T cells, especially the helper T cells, to activate a particular binding of antigen, and then we get something called a clonal expansion. So you get a lot of B cells that all express or will make the same type of antibody, and at first they might be making IgM, but eventually they'll switch over to either making IgA, IgE, or IgG, depending on what's necessary. And again, the IgE will be made so that will go and bind to mast cells. And then you'll end up with most of those plasma cells, which will be making antibodies of a particular type in response to something like a bacterial invasion. And then some will be converted into memory cells so that in the future, each response will be better. So that's a quick review of the immune system. Now let's talk about allergies. There actually people have classified allergies into 4 types. There's IgE-mediated, IgG, immune complex, and then cell-mediated. The cell-mediated type is actually broken up into 4 subtypes. So just to see how allergic reaction, how it all works together, you're going to have allergens, things that potentially are markers, basically, of the presence of something the body doesn't want to have present. Sometimes we intellectually might think, well, I don't want to be allergic, say, to pollen or to some drug, but the body thinks, doesn't rationalize this. It has cells, these antigen-presenting cells, such as the macrophages and the dendritic cells, and they will ingest the antigen. They will then go into the lymph node eventually. So they'll be floating around the body, take up the antigen, then go to the lymph cells. And then in the lymph nodes, they will take these antigens and we'll see they present it through these MHC molecules, major histocompatibility complex molecules, to T cells. And we'll see there are 2 types of T cells, cytotoxic T cells and helper T cells. And then that will lead to either the production of T cells, T helper cells, which will then educate the B cells to make certain antibodies, which will then go back and bind the antigen. Now, in the case if it's IgE antibodies that are made by the plasma cells, then of course they're going to go to the mast cells. And so it's not just that the antibodies are binding antigen, but they've actually then sensitized a particular cell, most importantly the mast cell, and then that will cause histamine release and the heparin release, and that will cause the symptoms that people normally associate with allergies. So it's the histamine that causes the 5 symptoms of allergic reactions: heat, pain, swelling, redness, and itchiness. And these all come from this mast cell. So much of what we normally associate with allergic reactions is actually the actions of these mast cells. So IgE, not a high prevalence antibody, but because it arms or sensitizes the mast cells, that makes it very important. So looking back to these different types of immunological classifications, we'll see the type 1 are IgE mediated, and they are all basically they're coating the mast cells, and that's how we get the reactions. Now, that's what most people think, all those skin tests and things are IgE, but there are these other types of immune reactions. So type 2 is actually mediated by cytotoxic T cells. This is, we'll see, is sort of most commonly encountered with blood transfusions and rejections of blood types. But they're not mediated by antibodies. They're mediated by cytotoxic T cells. Then we have a type 3, which basically we have antibodies link together and recognize antigen. And then the neutrophils, remember the neutrophils are the most common white blood cell type, will actually leave the blood vessels and go and engulf these. And basically they'll eat them up. Okay, and they will activate complement. Remember, so say you have an antibody attached to a bacteria, and then the complement comes, and it can go by using that C5b. It can recruit other complement elements, make pores in these bacteria, cause them to leak, lose their contents, and they die. Okay, and then we have the type 4, which are mediated either by the T helper cells or sometimes by T cytotoxic T cells. So there's actually an interplay that can happen here. These cytokines can happen, and they can bring in these macrophages, again, from the innate immune system. So there is this interaction between the adaptive immune system of making specialized antibodies or specialized T cells that can recognize antigens, and then the recruitment of other types of cells. But the T helper cells will work with the macrophages. The T cytotoxic T cells can directly bind to things and cause problems. There are some important diseases that happen which are not in the type 4 category, including type 1 diabetes. And rheumatoid arthritis, we'll see, is associated with a type 4 mechanism, and but it's also associated with other types too. So to bring this down, people actually in the type 4 is actually broken up into 4 categories. And even you can see this, something that you'll come across, Stevens-Johnson syndrome and toxic endoplasmosis. They have different mechanisms associated with them. So when it's thinking about treatments or what's causing them, it's important to understand what kind of subtype. You'll see here that we'll go through each of these types quickly, so you have macrophages get activated, just like in type 4A, just like happens with type 1. We see eosinophils, then we have the T cells, which get involved in type 4C, much like as happens with type 2, but they're different. But they both have cytotoxic T cells involved. And then the type 4, which includes what happens with type 1 diabetes, we see it's very complicated, but includes the recruitment of neutrophils and their involvement. So just quickly to go through, type 1, IgE mediated. You have degranulation is this process where the mast cells secrete histamine and heparin. Anaphylaxis, again, we looked at that. That's when you have the inflammation that causes severe swelling, say, of the airway, and you end up having trouble breathing. Then there's a type 2, which is there's something called the antibody-dependent cellular cytotoxicity, ADCC. Okay, and this again oftentimes happens with attacks on red blood cells. So it can lead to a hemolytic anemia where your red blood cells are bursting. And so actually, this is one of the mechanisms can be triggered by allergic reactions to sulfonamide and beta-lactam antibiotics. You can end up, one of the symptoms could be loss of red blood cells by this type 2 reaction. And even though T cells are involved, well, importantly T cells are involved, it's actually considered to be IgG mediated. So they end up recruiting elements that also attract the T cells. Then just to talk about this ADCC, because you will see this mentioned. So you have these natural killer cells, macrophages and neutrophils. So you have IgG antibodies bind and they target it, and then you end up with it being ingested by one of these. So in this case, the neutrophils are acting also to cause phagocytosis, or else the cells lyse, and they release different chemicals which are cytotoxic. And then from the attacked cell, you have antigens, which will be proteins and released. And then you can see it can end up being MHC1 will be associated with the cytotoxic T cells, or the antigens can be presented by MHC2, which is less the issue, and you'll end up having antibodies directed against the host in this case. This would be like an autoimmune reaction being triggered. So this this ADCC mechanism is basically can implicate both types of T cells. But when we normally think of the type 2 reaction, it's going to be more implicating the cytotoxic T cells. Then we have type 3, which is immune complex mediated. In this case, this is where we're talking again about antibodies binding to antigen, to then having the neutrophils come out. The complement tags them to be, see here 3B. Remember that causes macrophages to target them. The macrophages, in this case, are the neutrophils, which will actually leak out of the normal blood and go and attack it in the tissue. And then the type 4, which are mediated by cells. So these type 4 reactions are also called delayed type hypersensitivity reactions. Normally elements of the immune system, like complement, they're already designed to kill the organism. In the case of the delayed type hypersensitivity, it takes time for the T cells to become sensitized and then to make their way to the site of infection. So where a normal innate immune response can happen almost immediately, these cell-mediated types can take hours before there's a response, because it takes time for the message to get to the cells that need to then be kind of upgraded. So we can see here in this summary, again, different types of cells get involved with the different types of reactions. This type 4, it's the neutrophils are involved. And we see there are different types of presentation on the skin, but also these things can affect things inside the body which you can't see. So there's a lot, depending on the disease state, like with type 1 diabetes, you don't see that on the surface. Just a side note here, allergic reactions can come over time. So if you're interacting and having to gather information for someone who's having an allergic reaction, or expect if you have an allergic reaction, that just because you had it once in your life doesn't mean you're always going to have it. Sometimes it could be that you're always allergic, say, to strawberries, but oftentimes children will have allergic reactions to things such as food, which then over time will go away. Here we see, and this is actually from a study where they studied thousands of people, and we see here people between ages of 0 and 5 had a food allergy and it went away. But then later on, people develop things like nose allergies, in the linings of the nose, and that became more prevalent. Basically, runny nose, asthma type things coming in later. Okay, so that's why sometimes when you're older, you might, if you're allergic to something when you're younger, it doesn't always mean you're going to always have it, but you always have to be careful. Okay, anyway, immune responses change over time. So critical to immune responses is something called the major histocompatibility complexes, MHCs. And I'm mostly going to use this, but you will see that major, all mammals have MHCs. They have, if something has an adaptive immune system, then it's going to have an MHC. So this is the general term used. But you will see HLA used, and I'll point that out. So that's human or HLA, human leukocyte antigen, and that's specific to humans. So our proteins, the proteins that are actually MHCs, will oftentimes have the name HLA on them, and but do understand those are actually in this class. So we're talking generally MHCs because they apply to, people can study them in mice, people can study them in rabbits, and but they apply to people. But only humans have HLA molecules, so we'll see that. So anyway, so you have a peptide. Oh, sorry. So you have different ways that antigens get presented, processed, and then presented. So what's going to happen is this MHC is going to grab a piece of a peptide, and it's going to be what antibodies, in this case, it's going to be what either an antibody or we'll see cytotoxic T cell will recognize. So you have a long protein. It goes into something called a proteasome, which we're going to talk about in a moment, and it cuts it up into smaller peptides. It then will go to the endoplasmic reticulum, where it will bind one of these MHCs. That will then go to the peptide bound to the MHC will then go to the Golgi, which packages it into secretory vessels, which will bring it out to the surface of the cell, where then, if that antigen, a protein with that peptide in it, binds to it, it will then activate it. So this is one way. Well, actually, this is wrong. This is an antigen presenting cell. So a T cell is going to come, see this peptide, and it will then become the effector cell. The T cell, and if it's a cytotoxic T cell, will then be able to do the killing. So this whole cell here is actually the antigen presenting cell. And then the T cell receptor is on a T cell. If it's an MHC1, then it's on a cytotoxic T cell. If it's an MHC2, then it's on a helper T cell. And we see another indicator of a cytotoxic T cell is it has a molecule on it called CD8. If it's a helper T cell, it has a CD4. And we see that once the T cell has been exposed to antigens, and it need not just be one, okay, then it becomes mature. And so one of the things here, they use T with a small c for a cytotoxic T cell, and TH is actually a couple different types of, two main types of helper T cells. So it'll be TH1 or TH2. And these T cells, though, remember these guys are going to then be able to make something special. So just drive this home. Dendritic cell is one of the antigen presenting cells, much like macrophages. And actually much like a mast cell can also do this. It eats, say, the external thing. In this case, we're talking about bacteria. It then processes it inside of itself, presents itself on the surface to a T cell. So this is emphasizing in this pathway that these are not going to be activating cytotoxic T cells, but rather the helper T cells, because the helper T cells, we'll see other ones, that make the plasmas, that educate the plasma B cells and make antibodies. So this complex here we have the dendritic cell type. Again, like it's going to phagocytose. It's going to eat, say, an external thing like a bacteria. It's going to have different receptors, but always it's going to have an MHC complex. In this case, we see the CTLA4. Again, this is starting to. This actually can turn off this interaction, and there's actually drug targets we'll see for cancer that have to do with this in the future, and so you have this complex passing the recognition of this antigen onto this T cell, and depending, again, it's a cytotoxic T cell, it itself can go bind to something and attack it. If it's a helper cell, it's going to be making antibodies. And that's what we're going to see here. We have your antigen presenting cell, again, dendritic cell, macrophage, and actually B cells can can present to them, but we'll just let that be. So you're going to have this antigen presenting cell. It could activate a cytotoxic T cell, see the CD8. It could activate a helper T cell, see the CD4. Okay, and so depending if it's presenting that peptide in an MHC1 class molecule, it's going to do the cytotoxic cells. If it's an MHC2, then it does the helper cells. But the helper cells then can pass that antigen information on to B cells. Then the B cells can make more plasma cells, which make the antibodies. And we see that here, MHC1, we have class 1 here presenting. So that's going to be a cytotoxic T cell. And here, and again, here's the CD8. And then we have a CD4. These are on the T cells. And then we'll have that presenting. So this is how you get presentation of antigens. Very important. All these antigens that we're talking about at this point are peptides. So what's the difference between a MHC class 1 and an MHC class 2 molecules? And this is where we're starting to really build the argument for, or the understanding as to what can lead to drug allergies. So MHC class 1 molecules they have one insertion area into the domain. They have a alpha protein, which we call, and it has 3 lobes, and then it has a binding groove or cleft into which peptides can fit. And we see that down here. Peptide fits. And then notice there's this molecule called a B2 microglobulin. So this is not inserted into the membrane. It must associate with this molecule. If we compare this to the class 2s are actually made of 2 separate molecules that each have their own membrane insertion domain, and they have a bigger area. It's not one molecule, but 2 areas. So if we see these diagrams over here, we see that the MHC class 1s, we have one molecule and with a groove in it, and that's the antigen binding site. In the case of the MHC2s, it's 2 come together, and the binding site actually is sort of at the interface of the 2 molecules. So these are optimized for binding peptides. And an important point, ultimately a T cell receptor needs to come in and bind to elements on the MHC class 1 molecule and then form a molecular imprint of the antigens that are being presented. Not all presentations are productive. They do not all lead to passing on of the information, say, to the T helper cell. If the peptide is bound incorrectly, if there's something, if it's too big, if it's not trimmed right, if it's too small, and it keeps floating out of that, you're not going to get recognition by a T cell, and you're not going to get an immune response. So just because something is bound to a MHC molecule does not mean it's going to lead to antibodies or cytotoxic T cells that recognize that antigen. So this binding has to be well done. And I'll give you a little foreshadowing. So it's going to end up that this is the only mechanism by which the adaptive immune system can pass information on to make antibodies or cytotoxic T cells. So when it comes to drugs, there is no other class of molecules than the MHC molecules that can present it and have antibodies made against it. So somehow the drugs are going to have to integrate themselves into this presentation system. So you can just imagine if you have a drug and it's bound to this peptide and it's hanging over and it's too bulky or it has the wrong charges, you're not going to get antibody formation. It's going to relate to no immune response. So let me just bring up the importance of these MHC molecules. So I showed that MHC1s only make cytotoxic T cells. MHC2s work with the helper T cells. We mentioned that for the MHC1s, they can get their peptides from big proteins that are digested by proteosomes. So the MHC1 pathway, we call it the endogenous pathway. It actually gets its peptides, that are the antigens, from inside cells. So it's monitoring what's inside cells. Another astounding fact is that MHC1s are found on every cell in our body that has a nucleus, which is like all of our body cells, the somatic cells. So the MHC1s are the reason in the future, if you don't know, there's something called immunosurveillance. And if cells start developing towards being cancerous, proteins that are incorrect can be recognized on the surfaces of cells, and that will cause our immune system to react to them. So we have many cells that start to become cancerous, and because of this MHC1 endogenous pathway, and MHC1 involved endogenous pathway, our body can monitor all the cells in our body and detect whether or not they're presenting the right kinds of proteins. So MHC1, the fact that it occurs on all of our cells. That's not true for MHC2. So the exogenous pathway means that it can monitor things that are outside of cells. This is the specialty of the antigen-presenting cells. Those like especially the macrophages and the dendritic cells. And we see also that neutrophils can do this. They can eat things and then go through the MHC route, make antibodies. Remember now, MHC route doesn't make antibodies. It makes cytotoxic T cells. The MHC2 route makes the antibodies that we're most familiar with. So that's why it's important. So things come from the outside of the cell that can get phagocytosed. That's what's important. So let's look at this system again, which is involved with the MHC2. So you have the proteosomes for the which then will develop peptides to the endoplasmic reticulum where they'll load the MHC1s. And actually, this is what this is showing again. So inside the cell, they get ground up. There are certain proteins that are involved. The complexes, the MHC complexes, are not stable and before being loaded with a peptide. So there are other molecules associated with them called the loading complex. And then once they get bound to a peptide, then they can get freed, go through the Golgi, and then get presented on the surface. That's why I want to talk a little more about the proteasome. So proteosomes involved in many different types of activity. We're here concerned about the immune regulation, but they are basically like blenders, and they grind things up. Because they're so important in the cell, they have very many ways that they get regulated, whether from how many are expressed from genes to regulation of the complexes, and that's going to lead to a very important point. Proteosomes are basically made up of three parts. There's an internal part called the core 20 protein, and it gets two end caps. And so most of the cells in the body have a configuration where the end caps are the same. And so this is the system where proteins that are incorrectly made or basically have been worn out, they get charged with a compound called ubiquinone, which I don't have any slides on that here, but they get targeted by ubiquination, and then that makes them move to the proteosomes, which will then guide them up. And we'll see, you won't see in this course, but when you consider anti-cancer drugs, you'll see that there's a number that attack or target these proteosomes. If the proteosomes don't work, all sorts of basically all the waste builds up in the cell, and that targets the cells for attack by macrophages. So generally, proteosomes are cleaning things up in the cell, getting rid of the old proteins, kind of recycling them. Now, under the influence of cytokines, interferon gamma and tumor necrosis factor alpha, you will come across these names and proteins at some point. They're cytokines. They're things that can alter cells, and they can make something called an immunoproteasome. The immunoproteasome is found in the antigen-presenting cells. So what's in the dendritic cells and the macrophages is not the exact same proteasome as found in the rest of the cells in the body. And so these end caps are different. The core, there are different kinds of cores, and you'll see, so some is like trypsin-like activity, chymotrypsin-like activity. These are enzymes normally found in the gut for like digesting meat, but really we're talking about the degradation of proteins by these. And so they have different protein specificities. Here we even have beta-1, caspase-like activity. Caspase is involved in programmed cell death. And so remember I said, look at proteosomes are involved in a lot of things. Here we have apoptosis. Here we actually have a specialized kind of blender unit that would get kicked in and be expressed, made into proteins inside cells which are about to die. But here we have these different types, different endpoints, and we have this catalytic chamber. So it's kind of like a blender with different blending options. One can imagine for the immunoproteasomes that they will want to do a better job at grinding up proteins, and that's going to be the reason for that will be clear in a minute. We basically want shorter peptides to come out of this than one would maybe find in a regular cell. So the class 1 antigen, again, it will be derived from peptides that have been processed through the immunoproteasome, okay? And then it's going to, there's this protein called TAP, and it's part of a complex that stabilizes the MHC1 before it gets a peptide. And then it also then goes out and grabs peptides and puts it into the cleft in the MHC1. Now remember, the MHC1, the binding cleft, is made out of one protein. I should say, remember, I just mentioned that this MHC1 is found on practically all the cells in the body. And so each of the cells in our body are presenting about 10,000 peptides for checking by the immune system at any given time. So here on the upper left, we see the classic route where the peptides that are processed by the proteasome, they go through the regular route through the endoplasmic reticulum. Here's the endoplasmic reticulum, and the peptides come out, get trapped, and loaded onto the MHC1, which will then make its way through the Golgi to the surface. There's a reason people are interested in, is it ever that proteins made within the cell, could they ever have antibodies made against them? Because by this route, MHC1s don't trigger antibody production. But sometimes we observe proteins from in the cells and that there are antibodies made. So there's a big question, it's like, how does that happen? And this is going to be relevant because we're leading up here in this story to if a drug were to diffuse into a cell, how do antibodies get made against it? Because what I'm pointing out here is things from inside the cell go through this MHC class 1, and that doesn't lead to antibody production. So how could that happen? So that's why this slide is important. It's showing that actually the proteosomes can enter into phagosomes, which we'll talk about in a minute, which are a type of endosome, which is a vesicle within a cell. And they can actually then deliver it to MHC1 molecules there, or get loaded from the outside and in. There's different, people aren't sure, okay? But the whole point is that inside these vacuoles, MHC1 might be able to be loaded. And this could also be another mechanism as opposed to just the peptide going up to the lumen. The significant here is if if something can enter a phagosome, it's possible, because we're going to see that the MHC class 2 molecules could be in here, and that could lead to what we call crosstalk, cross-presentation. And so let's move on and talk about endosomes and phagosomes. So endosomes are naturally occurring processes where it naturally molecules that are presented on the surface of the cell, and a lot of times they're receptors. You heard the term receptor down regulation. That means maybe a cell had a lot of morphine receptors on it. And if it takes a lot of morphine, then you get receptor down regulation. It means there's fewer receptors on the surface. So how does that happen? Well, it can be that the cell makes less of those receptors, but it also can be, there's a mechanism by which things on the surface of the cell basically get dragged back into the cell, and they can be digested or they can be repurposed. And that system is the endosomes. So we have something, normally there are molecules called clathrin, and so you can have something called clathrin-coated pits. And you'll have like a receptor, like a muscarinic receptor, or I brought up the example of a morphine receptor. So it actually it pinches off into the cell, and it then forms a vesicle. And there are early stages, and later stages have more enzymes in them. And there's another type of endosome called the lysosome. And it has a lot of digestive enzymes and things that can cut up proteins, things that can oxidize things. So you can get drug processing in these lysosomes. And actually for some receptors, like if this is an MHC 1 or even 2 receptor, class 2 receptor, they can actually have the peptides that are bound in them swapped out. And then they can actually go back to the surface. So there's a way to do recycling. And that's what you see in the diagram to the right. You see there's endocytosis. You have these early endosomes. And they're taking other molecules. So what if that was a food molecule bound to the surface? What if that was like a vitamin? You know, they can enter the cell. They just don't diffuse in and float all around inside the cell. They start in these endosomes, and then the endosomes sort it. They send it to where it needs to go. They pair it up with the enzymes. They send it into different compartments in the cell. So you can, like in the case of food in the gut, the food, like we eat something, it doesn't just go from the inside of our intestine into our blood vessels. It actually first gets absorbed by the intestinal cells through endocytosis. And then, so in early endocytosis, then we'll see like a peptide coming in, and it will digest it maybe into its amino acids, and it will repackage it, and then it will send it out maybe, so it absorbed it from the gut, and now it sends it out into the blood vessel through transcytosis. So these endosomes are important for processing all sorts of things. And for recharging, in the case of the immune complexes, there's a thought that they can change what peptides are in there without having to go all the way through the endoplasmic reticulum. So this is important. It means that one could have a quick turnover of what antigens are being presented. So then we can go through late stages, and we see merger of these late endosomes with lysozymes, or maybe the lysozymes are actually evolved from the late endosomes, and then they can go the Golgi apparatus, which we'll talk about in a moment. There's a cis phase and a trans phase, and they package things up. So look at these phagosomes. So phagosomes are like endosomes, but they're specifically made. They're not just taking the cell surface. They're taking big things from outside the cell, like a whole bacteria, and they're taking it in and they're chewing it up. So in the case of antibacterial antibiotics, right, I'm going to keep repeating these kinds of concepts. If penicillin has reacted with something in a bacteria, and then the bacteria gets eaten by a phagosome, then the drug is going to get incorporated into the phagosome. And then it can go through these processes, and in this case, phagosomes have, like the dendritic cells and the macrophages, they present the peptides through an MHC class 2 molecule. And the MHC2s lead to antibody production. So you're starting to see that it's not that a drug for penicillin allergies, which we'll talk about, it's not that the penicillin diffuses into a eukaryotic cell, into our cells, and then we make antibodies against them. It's taken up by the bacteria, and then our cells eat the bacteria, and then they're perfectly situated to present it through MHC class II molecules such that T helper cells will see it, which will then present it to B cells, which will then have antibodies made against it. And that's the route. I'm going to be repeating it again. But that's why that antibacterial antibiotics are more likely to have antibodies made against them than to any drug that would just diffuse into a human cell. It's because eating things from the outside is more likely to cause antibody production than things coming from the inside. And I just emphasizing that the Golgi apparatus we hadn't really talked a lot about. But again, if the MHC class molecules are found in the endoplasmic reticulum, the rough one is the one that has ribosomes, so that means protein synthesis happens. So proteins get synthesized on the rough endoplasmic reticulum and then get packaged and processed through the Golgi. The Golgi kind of sorts faces. It has a face called a cis face, and it works through and can modify proteins. They can add sugars and other things to them if that's what needs to be added. And it puts them into vesicles, and here it shows it secreting it into the outside. But the main thing is it brings things to the surface. So if it's an MHC class molecule, then it gets to the surface, doesn't get excreted, it's still bound into the membrane. But it's brought the MHC that's now loaded with peptide from the endoplasmic reticulum through the Golgi and now presented on the cell surface. So there are some different thoughts. So again, back to this MHC1, that you could have it processed through the endoplasmic reticulum normally. But it could be that the antigen, if it's a peptide, could just be phagocytized. And all of a sudden, this is sort of the MHC2 pathway because it's in a phagosome. But if the cell surface, put it this way, when a bacteria comes in, it's pulling in also the cell surface membrane. And that cell surface membrane is also going to have MHC1 molecules on it. It's not the main thing one thinks of. But all of a sudden in this phagosome, there's MHC1 peptides, and they can get recharged. The original MHC1 peptide could dissociate from the receptor, and then a different peptide could be added to it, and then it comes back. So in this way, we kind of get presentation of different cells by the MHC1s due to this phagocytosis. This is a little more complicated. Don't worry about remembering any of this. So anyway, for the MHC2, similarly has a thing. You get phagocytosis because normally that's the pathway for the MHC2. It's things from outside the cell, and you're going to get some stuff caught that's on the cell surface will also get dragged into them, and you're going to have the antigens degraded. You're going to have early and late stages when things get presented to things. You're going to have travel through the Golgi, but then you're going to have this other way where it's thought that in the late phagosome, there's an actual thought that there's a little membrane piece, a vesicle basically again, that can then go back to the surface and present the MHC2. The idea is that you're going to need, at some point, you're going to have either an endosome or a phagosome involved, or the endoplasmic reticulum. Now, that up to now, I'm talking about how you get the peptides presented or processed for presentation. So the MHC class 1, remember, it was made up of one molecule. It ends up that it can only present peptides that are generally, it's optimized for presentation of actually peptides of 9 amino acids. Because it's one molecule, you see in this presentation, see it's all white, that shows it's one molecule. And so this one molecule just is less flexible. It has very stringent requirements. The MHC2, if you can remember, was made up of two different molecules. See, one is blue here, one is white, and here's the red antigenic peptide. And so it can actually accommodate peptides of 13 to 25 amino acids. These are much longer, and I'll give you a foreshadowing here. So if you have a drug and you want to fit it into either an MHC1 or an MHC2 cleft, you're going to have more possibilities than MHC2, because the drug is going to have to replace part of this peptide for the recognition site. And if it doesn't do it well, you're not going to have an immune response. So you have less space, less amino acids to interact with. So if MHC1 likes 9 and you got to remove 2 of them and replace it with your drug, then you're going to have less binding interactions potentially. That drug, that piece, better have some nice positive interactions. I mentioned that before being bound with peptide, these complexes aren't stable and they have helper molecules hold them together. If you replace some of these peptide pieces with a drug, and that drug doesn't also do a good job of binding, then the complex isn't going to stay together. It's not going to be good for presentation. The peptide's not going to stay bound to it as it gets processed by the Golgi and put into vesicles and brought to the surface. The MHC2s, it's bigger because there are 2 molecules coming together and it's kind of binding at the interface. One can see there's a lot more flexibility. There's a lot more potential. Look at the range, 13 to 25. So plus or minus 10 amino acids. One can imagine that 13 must be sufficient for binding. What if you then added another 3 amino acids worth of drug, and you're still fitting in there. So MHC2, and this I'll be bringing up now, is a better candidate for presenting drugs. And this then also, of course, leads to antibodies, which is, we saw with the type 1 allergic reactions, they're IgE antibodies. So you need antibodies to be made for many of the allergic reactions to happen. And of course, the MHC class 2 is what's important for making antibodies. And it also sees that it's more flexible. This does pose a little problem here, which I'll show though. People are talking about crossover of MHC1 to MHC2, or rather MHC2 to MHC1. The MHC1 peptides are kind of small for fitting into MHC2s. So in some ways, to expect crosstalk of an MHC2 going to MHC1 going to MHC2 doesn't seem likely because the peptide is too small. It likes 13 a minimum, and the MHC1 is like 9. Now, one can see going the other way, MHC2 to MHC1, because a longer peptide could always be trimmed a little bit and cut back down to 9. But you can't add amino acids to it. So the crosstalk that people are trying to figure out sometimes to explain different disease observations, it's actually harder to imagine it going from 1 to 2. One can imagine it going from 2 to 1. All right. These drug interactions. So what are we talking about here? Here's see HLA B27? This is a type of MHC molecule. So this is and that's what I'm trying to point out. So just because it says HLA, don't dismiss it. It's involved in the MHCs, and it's presenting an antigen. And so you can imagine binding pockets, different side chains, the amide groups are binding things on the HLA. And that means the flip side, they're presenting things that the T cell receptor recognizes. So if the optimum binding interactions, though, the stronger ones of which the MHC, one might imagine that actually the T cell receptor is recognizing basically the leftovers, what isn't binding well to the MHC. But in all cases, notice these systems are optimized for binding peptides, not proteins. So, but when we talk about drug allergies, we're talking about things that aren't proteins. And we see in this diagram, just to drive this home, that, I just want to show you. See, this is a diagram of, they examined a number of antigenic peptides that bind to MHC1, and they found that most of them were 9. Some of them went all the way up to 13. Remember, that's really important because 13 is the minimum size that could bind a MHC class 2. So there are some peptides that are presented by MHC class 1s which could also fit into MHC2s. But it's the minority. So if one had to expect cross-reactivity, like it being presented, an allergic reaction being caused by both an MHC1 and an MHC2, there's only a few antigens that can do that. The majority are not suited for cross-presentation. Particularly, again, 9 is the optimum length for an MHC1. And we see that here. Here's examples of the peptides. So a binding motif, amino acid, a leucine, or methionine, a valine. These are all lipophilic this is a lipophilic motif, and that's actually antibodies that are formed against HIV. We have plenty of antibodies against HIV. It's just none of them stop the disease. But as markers, it shows you have it. Here we have, and these are, and then we have another one. So this is the peptide. I'm sorry. So the lower one is the peptide bound by the MHC. The upper one is the motif that the antibodies make and recognize. So this is what it read. So you can imagine, here's a lysine that was interacting with the MHC, but the TCR receptor saw these ones in green. And the same thing goes here against influenza A. This is the peptide that was bound to the MHC, and this is what the T-cell receptor saw. And notice this can be spatial related. So antibodies can bind things also in 3 dimensions. And so their epitopes are not just limited to the linear sequence of the peptide. So in this case, though, you see, this is kind of like bottom one's what it's seeing, what's put into the system, and the top one is what comes out. And then here for class 2, notice a longer peptide, okay? And then we see that it binds this upper sequence. I'm sorry. In this case, these 2 are what's put in, and the green are showing what actually gets recognized. So in this case, for the transferrin receptor, the antibody is made to basically recognize one amino acid in that sequence. So this could get confused. This could be a non-discriminant antibody because many proteins have leucine. All right. So just to again show that from an antigen-presenting cell, so here we have a mature dendritic cell. It can present antigen to both class 1 and class 2 receptors, and then there are mechanisms so you can end up with production of cytotoxic T cells, or you can end up with production of antibodies against something that has been, say, phagocytosed. Okay, and this is just a simpler cartoon over here. So let's get into the drugs. Okay, sorry, this is getting to be long. So the drug allergies, we have all sorts of type. Penicillins can be both type 1 and type 2. So we can see it can both involve antibodies and it can involve cytotoxic T cells. And actually, if you look at this, penicillins are crazy. Also type 3s and 4s. So basically, we normally talk about penicillin allergies as just being type 1, but in fact, it can have a little bit of both. We'll see another class of antibiotics, sulfonamides. There's sulfonamide allergies. These aren't showing up as type 1. They're type 2 and type 4. And we see different allergic reactions and symptoms where, so for pulmonary drugs, bleomycin, methotrexate, these are anti-cancer drugs, procainamide, so isoniazid. These are, again, used to treat some bacterial infections. You're going to have ACE inhibitors, calcium channel blockers. These are regulating cardiovascular disease, yet you can end up with allergic reactions to them. But again, the sulfonamides, cephalosporins, much less so, which we'll be talking about in a moment. But best known are the penicillin allergies, followed by the sulfonamides. And we see they're associated with different disease states and different, And there's going to be a reason for that, which we'll make clear in a moment. All right, some definitions. I want to bring in, if you haven't been exposed to it elsewhere, the idea of a hapten. And then you'll have antigens, and then you have allergens. So to the common person and in your everyday life, you're exposed to allergens. Something's in the air. It causes you to have sneeze and cause you to have a runny nose. And that's an allergen, And you might say, in the spring and in the fall, there are different allergens, or people are taking Zyrtec or whatever, you're trying to stop an allergic reaction. More specifically, though, being more basic science oriented. So the allergens elicit an immune response, and that's what most people talk about. Scientifically, we're talking about antigens. So an antigen is a toxin or a foreign substance that can get an immune response. Okay, but specifically, a hapten is a particular molecule or a piece of a molecule that's actually leading to, say, the antibodies being formed against it, which is causing the allergic reaction. So most common people are not going to say, it's a hapten. But you as as pharmacists or whatever should understand a hapten is like the business part of the molecule. It's actually what the antibodies are made against. So if that hapten occurs, it doesn't matter what molecule it occurs in, it potentially could cause an allergy. And it might be that the antigen or the allergen, you got it from one place and you made antibodies against it, but the antibodies are made against the hapten, which is found in another molecule. And that molecule in and of itself isn't being presented by the MHCs to be made into antibodies, but it's cross-reacting. It's something that's recognized by antibodies that have been made against another molecule. So you got to be aware of that the patient history matters, what food, what drugs that they've been exposed to in the past, and not even drugs for the same disease. It matters what's the molecular detail, and that's the hapten. So most of the time we'll talk about antigens. Haptens are normally small pieces, just like what we saw before. We're talking about here recognizing a leucine and a valine. Okay? These are very small specific molecular features. So those are the haptens. Okay? So the antigen is the whole peptide. The hapten is the parts that it recognizes. So this leads to 3 models as to how the peptides get into the MHCs. So I just discussed the standard way of that it's going to get associated with the MHC in the endoplasmic reticulum and get presented. So in this case, this is the standard molecule. This is the best model normally. This is the one I think happens most. Is that a piece of the drug, normally a metabolite, gets covalently linked to the peptide. And then that gets presented by the MHC class 1 or 2. And that's this is nice because it's a covalent link, which means once it happens, it doesn't dissociate. And when we look at the penicillins, we will see that they absolutely, the things that are most allergenic about them is because they have a particular structure called a beta-lactam ring, which can be opened up and form a covalent linkage to peptides. And those are in bacteria. So this bacteria are going to eat the macrophages are going to eat the bacteria that contain the penicillin that's been bound to a protein. And so eventually it's going to make its way here. And so this is a very nice model. And again, this interaction would happen when the TPA takes it from the loading complex and adds it into the peptide inside the endoplasmic reticulum. But there's still the problem that sometimes some peptides seem to end up having antibodies made against them, or cytotoxic T cells, and they don't seem to, like they're coming from inside the cell, or they're somehow getting in there. So there are two models, additional models to the one where it's covalently bound. One is saying this PI model that it doesn't covalently bind to it, but it somehow associates with it. So one can imagine maybe the drug has a lipophilic piece and the peptide has a lipophilic piece, and you have van der Waals attractions, which then make them stick together. And this happens actually in this model, the PI model. It happens on the cell surface. That's what's key here. It's happening on the cell surface. So the drug never has to enter the cell. It just has to basically piggyback and somehow seep its way into the groove and associate it with the peptide and not disturb the T-cell MHC interaction. So this is a minimalist approach. What makes this, for this model, the hapten model, where it's covalently bound, that means the drug needs to have something that is chemically reactive. The drug or its metabolite needs to be able to react with the protein. So what if, so that would mean that you shouldn't have antibodies made against drugs with no chemically reactive units. But it does happen. And that's why this model can't explain 100% what's going on. It might explain 99% of the allergies, but not 100%. So that's why these other two models we're going to talk about matter. So in this PI model, the drug does not have to get processed into the cell. It just has to basically, at the last minute, before or while the MHC1 is sitting there waiting for a T cell to come along. Remember, so you can have 10,000 MHC. Well, if there's 10,000 on a regular cell, there's probably way more than 10,000 MHC molecules sitting on the outside of an antigen presenting cell. So they're not always bound with T cells, right? The T cells kind of have to go and survey them. They kind of come along, check out, oh, can I bind to this? No. And then they keep on coming forth. You can imagine there's an equilibrium. But if you have a T cell and it has to check out more than 10,000 possible interactions per cell, you have plenty of time for a drug to come along and also survey and see if any of the bound peptides could bind to it. And so if you have 10,000, if I gave you one possibility, I'd say that's not very probable. But if I'm talking about 10,000, if I'm talking about 50,000 possible peptides being presented, and none of them have a non-covalent interaction with a drug, all of a sudden the possibility of that happening increases. And so you could have a drug come and bind to one of the 100,000 molecules being presented, okay, and be lucky enough that when the T cell receptor comes to bind, that it still is in place and still makes a productive binding, okay. So it's a matter of odds, and so one can imagine. But then the thing is, how often can this be replicated? How often is a peptide presented that drug could interact with? Is it enough for? It might be enough for one time to activate one activate one T cell helper cell, which then makes some B clone B cells be activated, which makes antibodies. But then is that going to be long term? It seems to me that one thing going against this model is that of this causing a major allergy is that it seems to me it should be self-limiting, because yes, you might have antibodies made at one point in time, but it has to be consistently kind of made, in my quick thinking, so that it needs to be replicated. And so how easy is that to replicate? You might get other peptides recognized by the drug, but then they might make slightly different antibodies. It might not always be the same. It's like be worth a study. All right. Another way, this is the second alternative for non-covalent binding, is the drug actually gets into the cell. So imagine a drug diffuses into the cell, and and maybe it makes its way to the endoplasmic reticulum, and then so then you have the peptide loading complex, and the peptide needs to get loaded into the MHC class molecule, and then especially class 1 for the endoplasmic reticulum, and then it needs to bind there, and then it needs to make its way out, but it also goes through the Golgi. So I suppose the drug's in the Golgi, but you see the MHC class. So this is peptide bound to MHC, and MHC1, say, is not going to leave the endoplasmic reticulum unless it's bound to a peptide. But then it makes its way through the endosomes to go to the Golgi. It gets packaged by the Golgi and then gets re-put together. So the drug could bind to that peptide at any stage of the transportation process once the peptide is added. It could bind to it in the endoplasmic reticulum. It could bind to it in the Golgi. The idea is that the drug might be bound to the complex before it reaches the surface. And that's this altered peptide repertoire model. So there are these three models. This is my personal analysis. Okay. So again, the peptide one is the only one that has it covalently bound, the drug. The other two models, it's non-covalently bound. One model, PI, has the drug binding on the surface. The other one has it binding to the complex someplace inside the cell. There are problems here in that, say, if you have a groove, if the peptide is already nicely bound into the MHC molecule, where's the extra space for the molecule to fit? And that's a problem with the non-covalently bound ones, is that you've already had good binding. Where's there extra room for a molecule to fit? Okay, that's my thing. Okay. So the most likely model that's going to happen most of the time is the hapten-prohapten model. And it says prohapten here because sometimes you have antigen binding, and in the endoplasmic reticulum, there's actually enzymes that will cut it up and trim it. That's particularly necessary for the MHC ones, which have that optimal 9. So there might be sometimes it's if a 12 amino acid comes in, it might cut 2 off on one end, and that'll make one presentation, and then you take 2 or 3 off the other end, and that'll make another. So the same potential hapten or antigen could be presented in multiple ways, depending on how it's trimmed. All right. But in the end, always there's a peptide. That's if I ask a question, what's common between all the MHC molecules is they always have a peptin. There is no mechanism other than this for antigen presentation in humans. So there is no separate receptor that binds to drugs that then leads to drug allergies. The drugs and their metabolites need to somehow fit into this system. There's not a separate small molecule antigen presenting method. That's the main point. Done with the biology. Chemistry, quick. Sulfonamides. We'll be learning about sulfonamide antibiotics. We will see that these antibiotics have like a sulfur group in them that is doubly oxidized and attached to a nitrogen. So that'll be a sulfonamide. Okay. And antibodies form against these. They form parts of the molecule. Seems the antibodies, though, are formed against the sulfur part. So people with sulfonamide allergies sometimes report themselves being allergic to other drugs that contain sulfur. And so we have a meprazole, a protein pump inhibitor. We have aprenavir, which is a HIV protease inhibitor. We just have other classes of molecules that there's a drug allergy against them. And it is that people at first might have been exposed to a sulfonamide antibiotic and made allergies against it. It might be that, I said meprazole. Notice it's under here, non-cross-reactivity. So the antibody is not recognizing the sulfur with one oxygen, but it is recognizing the sulfur with two oxygens here. And it's not always that it needs to be attached to a nitrogen, but almost always. So it's normally the sulfonamide. A sulfate and this S. This is a sulfoxide thiol. So I misspoke there. No reaction against meprazole because the antibody is probably after sulfur 2 oxygens, not a sulfur 1 oxygen. And it probably needs the nitrogen. So, but the idea is that once you have antibodies, maybe against one class of drugs, you'll have allergic reactions against another. So just going by the class of drugs does not explain their allergy. Remember, the hapten is a substructure of the drug. It's not the whole drug, okay? There's an antibody made against a piece of it. And the question is, what piece is it made against to help you guide future therapeutic recommendations? So for penicillins, there are 4 kinds of penicillins. They have this ring called a beta-lactam ring. This can be opened, and it can lead to allergies. So penicillins have a 4-membered ring fused to a 5-membered ring. Cephalosporins have a 4-membered ring fused to a 6-membered ring. And so this is a big quandary, at least a lot of questions. If I have allergy to a penicillin, can I use a cephalosporin? Well, you can always just change your drug class and get out of the beta-lactams. But maybe for some pregnant women and other things, you don't want to do that. Okay, so then the question is, if this is penicillin, what is the antibody recognizing? All right, they have this beta-lactam ring, but we're going to see that that's not what the antibodies are made against, because the beta-lactam ring does not exist in the structures against which the antibodies are made. Now for penicillins, they are all going to have a same 5-membered ring. So if the antibodies are made against any structure in this 5-membered ring, like the gem-dimethyl group, okay, then those antibodies are going to are going to cause allergic reaction for anyone taking any kind of penicillin. But the side chains where this R is, they can differ. And oftentimes the antibodies made against the side chain, in which case then you got to check the structures for which side chains there are. You can get cross-reactivity, penicillins to cephalosporins, if they have the same side chains as the penicillins, which they do. And so you can see here, there are different generations of cephalosporins and penicillins. You see the frequency of types of hypersensitivity allergic reactions. There are different kinds. So highs, but serum sickness, basically having alterations in the blood due to taking one antibiotic when you're allergic, when it's had antibodies against another. And you see the percentages. So they can range anywhere from 3 to 11%. So, or itching, certain things are more tolerable than others. They have charts, and you can say, oh, if you've taken a penicillin, you should not take a cefoxetine, but maybe you could take another one. And all of this ends up going to be structurally related. You can expand that chart, and you can see different generations, and then you can see what might be okay to take and what not. Again, the experts in pharmacy practice will tell you how they think you should approach this, but and people have different opinions about how you approach this. But you can see that even within the cephalosporins, there's certain of them, this is just based on antibodies made against the side chain, that if you were to have a reaction against a cephalosporin, which is much less common than penicillins, that it might not be driven because of a penicillin reaction. It's basically driven against the hapten. What is the exact group recognized by the antibody that's responsible for the allergic reaction? So the penicillin reaction, most common, skin tests. And this is what's important. With penicillin, and this is something we could talk about that could be considered for other drugs. It's actually best known for penicillin. But this is the thing. There's something called the major determinant, antigenic determinant, and the minor antigenic determinant. The major antigenic determinant, if you notice here, has a protein that has opened up the beta-lactam ring. And so again, you can imagine this being in a bacteria. The penicillin's gotten in. It's doing its job. And then a bacterial protein, actually, maybe a protein, actually the mechanism of action you'll see among them is a suicide inhibition reaction of the penicillin with a D-alanine transpeptidase. So imagine this protein being the D-alanine transpeptidase, which is the mechanism of action for the penicillin, one of them. So that gets made. And then the bacteria gets eaten by an antigen-presenting cell, and it gets processed. And then that protein with the penicillin attached gets loaded onto an MHC class II receptor. And then that MHC class II receptor gets presented to a TC helper cell, which then passes the information on to a B cell, which then gets antibodies made. And we end up with a penicillin reaction. That's why this is critical. Now, here they just show, well, the less determinants are other forms of penicillin, and they're kind of showing it reacting with the T cell. Different people have different thoughts on how this works, but here we have different things, and we will learn about penicillins is that they're not stable. They're very reactive, but you can see that there's different ways and different proteins could react with the beta-lactam ring. So while the previous slide didn't show the minor determinants also being reacted with a protein, and actually that probably does happen. So this is following that first model, the hapten one, which is the most likely to be successful. And here we see penicillins are very well suited to covalently interacting with bacterial proteins, and then the bacterial proteins being ingested, chopped up. And I'll tell you something, bacterial proteins are very immunogenic. They have amino acids. They have all of our amino acids and all our proteins are L. And bacteria mix them up. And so when a D amino acid is presented, it's going to be seen that it's something from outside. It's something that an antibody should be made against. And if we were to study vaccines at this point, you'd see there's things called that the more you can irritate the immune system, we call them adjuvants, and the bacterial protein could act as an adjuvant. It could actually enhance the immune response, the recognition of the T cell by the peptide with bound drug and by the MHC cell. So here we see, again, in a discussion on penicillin allergies, you have the commonality of the beta-lactam ring. But what I'm saying is that it's not expected that the antibodies recognize an intact beta-lactam ring. They will, if anything, recognize altered forms, opened up forms, rearranged forms of the beta-lactam ring. And so you're going to have the major determinant definitely bound to protein, and you're going to have minor determinants. One can argue maybe this is following the other. The minor determinants could be trying to use the other pathways, which would be non-covalent interactions, which would be much rarer. So therefore you'd have less likelihood of reaction. Hence there would be minor determinants. But here, this particular example up here has an intact beta-lactam ring. But that would be very less likely to happen. But you see, these are other possibilities: an open beta-lactam ring, a rearranged beta-lactam ring. But the major one, the most likely cause of an allergy, is the protein. Which this is what happens to the D-alanine transpeptidase. This is mechanism of action would then be causing the allergy. So we're getting close to that last slide here. I know we've been going on forever. Here we have cephalosporin allergies, and we see that, if you're allergic to, say, ceftriaxone, you probably shouldn't then give cefuroxime and ceftazidime. And so we see that if the hapten here, this is a 5-membered ring, amino sulfide containing ring. Here's a furan ring, which is a aromatized 5-membered ring with an oxygen. Or here, group 1 and 3, if the antibody is made against this hapten, then we can understand it maybe would hit group 3. But why group 2? And here you see it's because they share this methoxyamine group in common. So if the hapten, if the antibody is made to recognize that group, then you could have cross-reactivity. If it's made, say, to react this group, then you could still give cefuroxime. But nobody tests. You can't test what hapten the antibodies are causing the reaction. So you just wouldn't give it because you don't know. But if you don't mind putting a person's life at risk, you could give them each of the drugs from each of these classes and see their reactions. And if they have a reaction against one from group 1 and group 3 and not group 2, then you're by deduction, you can assume the reaction might be against this heterocyclic ring and not against this hydroxyamine. Would you ever do that? No. But scientifically, you could. So, importantly, these are the final notes. It's important what the structure of the drug is, not the use of the drug. So it's not that people are allergic to sulfonamide antibiotics. They're allergic to the sulfonamide group in those drugs. It's not that they're allergic to all beta-lactams. No, they're allergic to maybe the side chains in those drugs, or some other structural feature. The likelihood that someone's going to be allergic to all beta-lactam antibiotics is very low. Okay, so you have to think. But now it's why it's important to make a guess as to what the haptens are, is because that's basically the trigger, right? If you eat that hapten, then you're going to have an allergic reaction. And so if you can make a guess what it could be, then you could just avoid it, and then you wouldn't have allergic reactions. And also, the first time someone has an allergic reaction, they must have been exposed to that hapten before. So sometimes it's very confusing, like, why does somebody have an allergic reaction to a particular drug? And that's where drug history comes in. For example, penicillamine basically has the structure of the groups in the 5-membered ring on all penicillins. Penicillamine itself can be used to detoxify heavy metals. It has no antibiotic activity, but it has the exact structure. So almost nobody would be given penicillamine for treatment, but if they are, and that was in their patient history, then you should be reluctant, or you should be very observant if you're going to give them a penicillin, because they have been exposed to that structure. And so one can only speculate that if someone has an allergic reaction the first time they're given a drug, that they must have been given another drug previously or eaten something that contains a chemical that contains a similar structure. To recap, we don't know from the endogenous pathway that goes to MHC class 1. From the exogenous pathways, that goes to MHC class 2. Okay, that that normally you can have phagosomes that can transport it, or through the endoplasmic reticulum and the Golgi, the MHC class molecules charged with peptide can make it to the surface. Where the peptides, where do drug metabolites get incorporated into this? Is it the drug? It's simple if it's coming from phagocytes. being eaten from bacteria. But what about any other kind of drug? What if the drug is penetrating? So antiviral drugs, we'll see. It's not that it's not our cells are eating the virus. The viruses are getting into our cells. So and then that means that drugs get inside the cells. So how do we make antibodies against the virus? How do we do that? So normally the drugs will go in and act on the virus, say. But if we're going to have an allergy to the drug used to treat that, that means the drugs are designed to get into the cell. And then at what point? Do they covalently interact with the peptide, or do they non-covalently, that third model, interact with it? Is it in a phagosome? Is it in a proteosome? The proteosome is not going to have any interaction. But, we don't know, okay? We don't know exactly when, and that might influence what properties of a drug could we ever predict that could be antigenic or not. So there's stuff we don't know. Let me tell you one thing. What if we wanted to make antibodies against the drug? People do want to do that for testing. Like if you want to test the blood levels of a drug, one could make antibodies against it. People don't because it's too hard. So in general, though, again, you could take something from the outside, like a drug, and inject it into an animal, and then through using those MHC2s and the T helper cells, you could end up with IgG and IgM antibodies being made. There is a change over time. IgMs are quick recognizers, and then they switch over, remember, through that B clonal expansion to IgGs. And that we see that the binding ability of the antibodies, especially the IgGs, improves over time, where that isn't with the IgMs. So what about, you can actually, there are some companies that contract out to try and make antibodies against drugs. If you take small molecule, this is one company's strategy. If you take the drugs and you inject it in a rabbit, you don't get any antibodies. This is to be expected. So most drugs will not make antibodies. But here's the thing. If they pre-react the drugs with like serum albumin, a protein, and then inject it, then they get antibodies. At this point, you should be able to understand why this is so. Because in the first case, the drugs may never interact with proteins. And if they're not suited somehow for that second model where they could somehow sneak themselves in and non-covalently interact, I say that's probably a rare occurrence. But it has a possibility. But the binding to a protein, which would then be phagocytized and presented, that's very low, to the point where you don't expect antibodies to be made against drugs. But if you pre-made the complexes of protein, then the proteins, right, would be phagocytized, and then eventually you'd get presentation by the MHC2s, and then you'd have antibodies made. So that's the way you purposefully conjugate. If you wanted to make antibodies, you purposely conjugate them to protein. Putting it all together. So, antibacterial, this just recaps the thing. Drugs given to bacteria, which then have a chance to interact with bacterial proteins, have a highest chance of being made, having antibodies made against them. And again, bringing up that point that bacterial proteins are immunogenic. Our immune system recognizes the wrong amino acids and it makes antibodies against them. If you tag a drug in there, that's not a bad thing. Viruses inside cells, it's harder for drugs that are already inside of a cell to associate with an MHC class molecule and then present it on the surface. So it's harder for drugs that get absorbed to actually have allergies made against them. Drugs that bind to the cell surface could be endocytosed, and basically that's kind of like phagocytosis, and then they would be more likely to have drugs or antibodies made against them. And then the allergic symptoms will give some hint as to what kind of process the drug experienced in order to become allergenic. So if we go back and look at the types of allergies observed, kind of close the circle here, we see that the type 1 reactions are going to be all MHC2 mediated. They're going to involve the making of IgE antibodies, and they're going to be coating the mast cells. So for these types of type 1s are going to be particularly suited for any allergens coming from outside of our cells. And so that's going to mean that they're going to come from drugs. If we're talking about drug allergies, we're talking about drugs that have interacted with things like if you're treating bacteria, if you're treating worms, if you're treating fungal infections, all those are things that are going to be eaten by mast cells and are going to be making antibodies, especially the IgE, okay? You can get IgG made, okay? So that's why we see, if we look at here, beta-lactams are both under type 1 and type 2 here. And so you're going to get some IgGs made. That does imply MHC2, okay? But this can also, I'll leave it at that. This can also involve cytotoxic T cells. This particular diagram is not really emphasizing that. But again, the reactions with red blood cells can bring in cytotoxic T cells. For some reason, this isn't showing that so well. And then we see that most things are mediated by MHC class 2, except for the type 4C, which specifically is related to the cytotoxic T cells, and that includes hepatitis. Oh, and finally, not quite, remember, I want you to remember this. The MHC1s are made, the binding site is made up of one molecule, binds amino acids of 8 to 10 most of the time, with 9 being optimal. The MHC2 is able to accommodate larger molecules, 13 to 25 amino acids. And so I'll probably ask you this, because this gives indication of the size of the cleft. And again, the MHC is made up of 2 different molecules, so it's much more adaptive and flexible. Because these can spread apart a bit, you can just imagine more flexibility with a larger binding area. And so one should expect that MHC molecules, which of course lead to antibody productions, can better accommodate drug molecules. And that will be the reason for most drug allergies. So putting it all together, here's that slide that we began with. Here, we want to see then that we know we're going to have an MHC class molecule. It's going to have a peptide of some length bound to it. And somehow, whether covalently or non-covalently, a drug will have to associate with that peptide and then be recognized for a T cell. And if any of these interactions are wrong, what if after all these amino acids are bound, the place where the drug binds puts it here so it can be recognized with a T cell. And remember, the T cells are going around, it's checking tens of thousands of presented peptides, trying to find one that it fits to. And it comes along, and it happens to have a bunch of valines here, which are very lipophilic. And it ends up on the drug, there's a bunch of charges. Well, this isn't going to work. This is going to destabilize the interaction. And you might not get T cell binding, and we'll have to go check out another peptide. But then that peptide might not have drug bound to it. So there's some probability going on here and ability. So a drug can interact with a protein. It doesn't mean that protein, when it binds to an MHC, is going to end up producing a productive interaction with a T cell. And so just because a drug binds to and interacts with a protein does not mean you're going to develop drug allergies, okay? And there's a lot of factors that have to be correct in order for these T cells to be activated, say to either make antibodies if they're T helper cells, or to make their own receptors on their surface if they're cytotoxic T cells that would then recognize something. And this is the end. It's a lot harder than general chemistry. And if you have any questions, I hope you can go back, review the slides if there's some things that weren't clear. And I know this is a long presentation, but it covers everything you need to know not everything, but what we know about drug interactions or with allergies. Thank you.